Okay, so basically we started yesterday a very important topic from this chapter 6 that's called as protein synthesis and nucleic acid, and that chapter have a very important topic that is called as that how we can say that uh, there is a semi-conservative kind of DNA application. So how we are going to find this out? We have basically very important these two names. You need to remember them. That was Matthew Melson and Franklin Straw. So we also used to use their last names, and we used to term them as Slaw and Melson. Experiment. That's going to tell you the conservation model of this E. coli was used in it. That's a bacteria which is present in your gut all the time. And I hope you remember we discussed in the last class that we are going to basically take these two E. coli in two different mediums or sources, in which one we used N15, that was a heavier isotope, and in other we basically allowed them to divide and divide for many generations. So first of all, step one was that we allowed this E. coli in the nutrient material, which was containing the heavier isotope of nitrogen. And then we shifted after many generations into the next beaker, which was containing this N14. Okay, that was the lighter isotope. And as you already know, I have already told you that your bacteria is going to replicate or divide by binary fission after every 20 to 30 minutes. So it means that after many generations, we can use this bacteria for this experiment. So what they did, we discussed yesterday that I'm just quickly swiping those slides which we have already done in the previous lecture. So this is the same thing, but here I would like you to remind one important thing, and that is the use of this centrifuge machine. I hope you remember we discussed that. It's called as centrifuge machine, and it's very much important for basically changing, or you can say that for extraction of the organelles. And one principle for this centrifuge machine is that denser will always, whatever the denser thing will, it will always sink first. Okay, so on this basis, we are basically revolving or spinning this centrifuge machine, and we use the samples in a test tube, and then we extracted from both the heavier and lighter isotope DNA. And at the last, I'm just going to repeat again that the heavier DNA that was closer to the bottom first, and it came to rest first. Obviously, the heavier one was this. Okay, so we discussed here in the last lecture, and then we wanted to discuss from here. So have a look again. Let's revise again the all the conclusion of that how we can say that your DNA is replicating semi-conservatively. So the first step was that bacteria was grown in N14 medium over here, and the second, you tried to grow them in different medium. That was the heavier one, means N15 medium, and then you shifted them from lighter to heavier or from heavier to lighter. You can do it both. Like, we basically want to compare the two. So bacteria are transferred to N14 medium, and then samples were removed at different intervals. So it's it can be N15 here and it can be N14 over here. It was first grown in heavier, and then it was transferred to the lighter one. And what we did, we basically after generation, after 2 generations, after 3 generations, we basically extracted the DNA from the bacteria, and we know that this is called as plasmid. That's a ring-shaped separate DNA in different bacteria, which are also used in biotechnology and even for the production of insulin as well. I hope you remember from your O levels. Okay, so the extracted DNA was basically put in this test tube, which will be run in the centrifuge machine. Here you can see that we are showing you the assemblage of the centrifuge machine. So this DNA suspension was basically run, you can say we use the word RPM, that's called as revolution per minute. And what will happen, the heavier will obviously rest first at the bottom. So here you have a look. We had, in one test tube that was control having lighter isotope, and the parent which was having basically the strands which were having the heavier isotope, And the third generation, which we got, that was the F1 generation. When we started that DNA, we came to know importantly one thing, that one of these strand, if I'm showing you like this, one of these strand was from the parent and there was a newly synthesized strand over there as well. So it kind of giving us a hybrid, one was N15, other was N14 strand, and this was giving us a hybrid DNA molecule. Okay, that was on the F1 generation. You need to remember it. And then we allowed this to replicate further for F2 generation. It's for you, I think you remember from the genetics chapter, but it's called as